Hi guys, today we're going to be going over USACO Bovine Genomics. So in this problem, Farmer John has N cows with spots and N cows without spots. And so basically, Farmer John sequences the genomes of his cows, and each genome takes up a string of length M, and it's built from A, C, G, and T, right? And when he lines up the genomes of his cows, he gets a table, as we can see here. And for this table, n equals 3, so there are 3 cows with spots and without. And so looking at this table, he surmises that position 2 is a possible location in the genome that could explain spottiness. And this is because basically Farmer John can predict which of the cows are spotty and which are not here, because all the plain cows have C while the spotty cows have A and G. So we can predict with a pretty high amount of confidence that C determines that the cow will be plain and not spotty. And so position 1, for example, is not sufficient by itself to explain spottiness, since A in this position might indicate a spotty cow or a plain cow, because notice how A is in both. Okay, so given the genomes of Farmer John's cows, we have to count the number of locations that could potentially, by themselves, explain spottiness. And for our input format, it's not going to be pretty like this. We're just going to get n and m, and then the next n lines contain a string of m characters, and that's going to describe the genomes of the spotty cows, and then the final n lines are going to describe the genome of the plain cows. So we have 3 here and then 3 here, right? First 3 are going to do spotty, and then the last 3 are going to do plain. And for our output, we just need to output the number of positions in the genome that could potentially explain spottiness. And a location potentially explains spottiness if the spottiness trait can be predicted with perfect accuracy among Farmer John's population of cows by looking at just this one location in the genome. So basically, we have to be 100% sure that a specific location will prove to be the location where um, the spottiness could possibly be linked to. So basically, um, all we're looking for is that there are no duplicates. So for example, like we could have A and G here, and then we could have like C and T here, right? But as long as the traits don't overlap, so as long as these letters aren't shown here and these letters aren't shown here, that's really all we need to prove that that could be this that could be the specific number in order to predict the spottiness. And remember, we don't need to output the number, we just need to output however many spaces can predict the spottiness with 100% accuracy. So with that, let's move on to our programming portion of the video. Okay, so in our program, we have some pretty straightforward stuff. We have our static variables, we have our main method, we set up our in and out, call the methods, and close our in and out. And remember, our initialize method sets up our static variables, and our solve method actually solves the problem. So in our initialize method, we set up n and m, and then we initialize spots and planes. And then here we set up spots and planes by filling them in, right, with the genomes. And in our solve method, what we're going to do is first we have to make our count variable, which is going to store our answer. And then here we are iterating through every single genome. So we're going to take the first letter of all of the genomes, right? All of them, including the spotty and plain. And we're going to set up 2 hash sets. All you need to know about hash sets, if you don't know anything about them, is that they are kind of like array lists, except they don't allow duplicates. And so basically, what we're going to do next is we have a Boolean called add, which is equal to true. And the add variable is basically just going to determine whether we add one to our count or whether we don't. So whether it qualifies as the determining factor or the determining spot in the genome. So what we do is we set up our hash sets with these 2 for loops. What we do is we're going to add spots in position B, char at A. So what this is going to do is it's going to go through, this for loop is going to go through every single genome that we have in the spotty category. So if you look at the example that we're given in the problem, you can just scroll back to it in the video. But basically in that example, we have an N of 3. So basically we're going to take in 3 Ns, or sorry, 3 genomes, and we're just going to add the character at the Bth um, Bth position, right? And it's going to be the character at the Ath position. Right now A is 0, so it's going to take the first character. So basically all this does is it just takes the first character of every single spotty cow's genome, and it's going to add it to the hash set. And in the hash set, remember there's no duplicates, so if there are like AAG, then in the hash set it's just going to be A and G. We do the same thing for the plane, right? Pretty much the exact same thing. And then here what we're going to do is we're going to do a for each loop that goes through spotty and also plane. And so what happens here is we're going to take the first character in spotty, right? Let's say that it's A, okay? And then we're going to take the characters in plane. Let's say that the in plane right now in the hash set, let's say that we have C and T, okay? So I'm just going to add comments to simplify it. We have A in spotty right now, and we have C and T. I'm not sure if those are the accurate, but we'll just pretend that they are. So let's say that we have A in spotty and C and T in plane, and if SP, right, spotty is equal to plane, PL, then we're going to make add false and we're going to break. What this means is that right, if we break, that just means that we have two characters that are in both the spotty and the plane, and that makes it so that it's invalid. It's not going to be able to determine if it's spotty or plane, because if the gene is in both of the categories, then it really has no use. So in this case, let's run through the for loop. We first have A, and then we first have C. We check, is A equal to C? No, it's not. So we keep going. Is A equal to T? No, it's not, right? So because we've gone through this for loop and we haven't done this a single time, add is still going to be true, and we're going to add one to our count. And finally, we print out our count. So basically, this is just the code version of what I said verbally earlier. It's just checking whether there are any sort of duplicates between the spotty and the plane. And if there are, we don't add it. If there are no duplicates, then we do add it to our count. And so now we are ready to submit. And as you guys can see, we pass all the 10 test cases. And so that's basically it for Bovine Genomics. If you have any questions, feel free to leave them in the comments and I'll try my best to answer them. And just thank you for watching to the end of the video. And I will see you guys in the next one.